Good afternoon, everyone. On behalf of Sartorius, I would like to welcome you all to the webinar titled Unlocking Potential: Seamless Integration of Cutting-Edge Digital Solutions in our CLD Platform. I am Abhishek, a conference producer at IMAPAC and your host for the day. I would like to share a little bit about us. IMAPAC strives to be your one-stop shop for the biopharma industry's business intelligence and networking opportunities. Over the past 15 years, we have established ourselves firmly as a trusted organizer of high-quality biopharma B2B conferences and exhibitions in Asia Pacific and beyond. In the recent 2 years, we have exponentially expanded our portfolio to include digital marketing solutions, market research reports, and our webinar series. Today, we are hosting the Unlocking Potential: Seamless Integration of Cutting-Edge Digital Solutions in our CLD Platform webinar powered by Sartorius. Here's a quick introduction on Sartorius. Sartorius is a leading international partner of the biopharmaceutical industry and the research sector. Known for its innovative solutions, Sartorius specializes in providing cutting-edge laboratory and bioprocessing equipment, as well as services that enhance the development and manufacturing of pharmaceuticals. With a strong commitment to quality and sustainability, Sartorius supports its clients in making scientific advancements that improve human health. I would like to express my heartfelt gratitude to Sartorius and our distinguished speaker for making this webinar possible. Thank you so much for your time and commitment to be a part of this webinar. And to all attendees, thank you for taking the time out to be with us here today. Before we start, here are a few housekeeping announcements. We strongly encourage everyone to submit your questions using the Q&A function at the bottom of your screen. The representatives from Sartorius will be answering them during the live Q&A session after the presentation. So now, without further ado, I would like to introduce our speaker for today, Dr. Yash Patel, Product Specialist, Cell Line Development at Sartorius. For introductions, Dr. Yash Patel joined Sartorius in August 2021 as a Product Specialist in Cell Line Development. He has a PhD in Chemical and Biological Engineering from the University of Sheffield in collaboration with AstraZeneca. Dr. Patel has 14 years of CHO cell line development and engineering experience with CDMOs such as Lonza, as well as in academia. He is an author of numerous publications and a patent within the field. Dr. Yash, the floor is now yours.
Thank you very much for the kind introduction and giving me the opportunity to speak here today. So as mentioned, my talk is Unlocking Potential: Seamless Integration of Cutting-Edge Digital Solutions in our CLD Platform. And what the talk will encompass is, firstly, I'll set the scene a little bit, and then I'll introduce our 4Cell CHO platform and our CLD service, and then touch on some of the digital solutions which we've implemented in the platform in the form of, firstly, automation to accelerate the CLD process, and then a predictive model using machine learning to help de-risk the CLD program. And finally, then I'll summarize my talk and then be happy to answer any questions which may come up during the presentation. So let's get right into it. As you know, and as we all know, the drug development journey is quite a lengthy process. It has a range of different steps along it and a lot of activities to be performed. For instance, you go from drug development to process development and eventually into drug manufacturing across the different clinical phases, from preclinical to phase 1, 2, 3, and eventually into commercialization. And during the journey, you have to do a number of key steps which can be highlighted here below. And as we know, from discovery to commercialization, the drug development time can range from 8 to 10 years. That can be faster and can be longer depending on the individual molecules, but it's still quite a lengthy process. So one of the key challenges which we've identified is that to develop a drug, a key factor is to be able to get it to the clinics or to the market as fast as possible. And this is a very important factor when you're in a competitive environment where there's a range of different modalities and drugs being developed at the same time. But a key aspect during the CMC process and when you're going through this drug development process is identifying your optimal cell line. And the cell line has a crucial influence into the CMC process. For example, a high titer cell line can affect your cost of goods and make them lower, A optimal CQA profile can make your drug very efficacious, which is also very important when it comes to the clinics and actually the effectiveness of your therapy. So the cell line has a key influence over the CMC process and also ultimately what is then your manufactured drug substance or drug product, which you're going to inoculate into your patients or administer to your patients. So with the cell line, a key aspect you need to think about is being able to still maintain that robust selection and be able to identify that optimal clone, but do it as fast as possible. So how have we sort of addressed that key challenge? And that solution, particularly through digital solutions, is twofold. One, we've introduced automation, which allows us to essentially hone the process and be able to identify those high producing clones and select them in an accurate manner. And then secondly, with the predictive model, and using elements of machine learning in the model, we've been able to strengthen our selection process to be able to identify that optimal clone. But what are the values? What value does this add to your process? Well, or do these solutions add to the process? Well, automation essentially has we've shown to has accelerated our CLD process. So essentially, you get to identifying that lead clone or getting to that clone quicker, whereas the predictive model has de-risked the CLD program and it ensures that you identify that optimal clone more effectively. So with that challenge, I've sort of set the scene, and I'll go into in the following slides into a bit more detail, essentially, of how we've implemented automation and the predictive model. But before I get into that, firstly, I thought I'd introduce our platform, our service, and our workflow of how we do it, and how the automation and the model sits within our CLD workflow. So let's go right into that. So firstly, an introduction of our platform. Sartorius's 4-Cell CHO platform is made up of 4 key components, of 4 key individual services. Firstly, we have our own proprietary cell line development platform, which essentially goes from DNA to RCB in 9 weeks and can achieve titers up to 10 grams per liter. Now, associated and coupled with the cell line development is our protein characterization capabilities, where we can perform characterization of the molecule in support of the cell line development to identify that optimal lead clone, but also then to continue the support through the entire CMC process. Once you've identified that lead clone, we can also manufacture the master and working cell bank through our cell bank manufacturing facilities in Glasgow, and where our facilities are fully GMP qualified, and we can manufacture up to 500 vials at 12 million cells per vial. Now, with any master or working cell bank which you manufacture, you need to perform the necessary biosafety testing to release the banks. And there we have the full suite of validated GMP release assays, including sterility, mycoplasma, virus testing, and also the genetic characterization. Now, encompassing all 4 of these services is also our proprietary 4-Cell Smart CHO media system, which is essentially a ready-to-use, off-the-shelf media system and supports scales from bench to manufacturing as well. So let's go into a little bit more detail of our CLD platform. It's made up of 3 key components. The first component is our own proprietary CHO DG44 host cell line, which is a suspension growth cell line and has a fast doubling time, approximately 18 hours. This cell line has a full cell line history report, which is documented and in accordance with ICH and WHO requirements. Coupled with your host cell line, you need to have an optimized expression vector. And we've developed our own proprietary expression vector system, which can support expression of single chain molecules, 2 double chain molecules, and also in a combination multi-chain molecules. And this optimized expression vector has been developed to work synergistically with the host cell line and has all the necessary genetic key components to really have optimal expression. One key factor to mention with our platform is that it is freedom to operate. And what that means is that we have no licensing fees or royalties associated with our platform. We're a fee-for-service based model when it comes to generating cell lines with our partners or for our clients. Now, coupled with your host cell line and your expression vector is that you need to have an effective media system. And there we've developed Smart CHO media system, which is optimized for our host cell line, is chemically defined, animal component free, and supports those high cell densities, viabilities, and productivities which are necessary for that lead clone as you go through to the CMC process or the remainder of the CMC process. The third key component in our platform is that Sartorius isn't a CDMO. We do not do GMP manufacturing, but we do want to support our clients as they transition into that phase. And the way we've done that is we've developed an optimized upstream process design, which has shown scalability up to 2,000 liters. And what that supports is that it supports our clients to be able to transition into the remainder of the CMC process or into process development and GMP manufacturing, either on their site or with the CDMO of their choice or the CDMO partner, in a seamless manner. So that's a snapshot of our platform. But our platform has constantly being evolved or evolving. And when Sartorius acquired Celco in 2015, but before that, when we were Celca, this platform was originally developed. And this platform started in commercialization in around 2010, 2011 with version 1, which had our DG44 host cell line and used DHFR as our selection system, which is a very common selection process or expression system used within the industry. We used FACS for single cell cloning, and we developed our first version of our upstream process design. With version 2, we started to expand our platform's capabilities and versatility towards expressing different molecule types, not just monoclonal antibodies, but expanding that. And I'll go into a little bit more detail of our track record of what different types of molecules we have expressed in the following slide. Continuing on to version 3, we made a key improvement by removing MTX in the selection process. And the reason we did that is that we appreciate that MTX is a toxic chemical. Additionally, with the gene amplification capabilities, it does also associate some genetic instability. So what we did is we developed a process where we removed the MTX, but maintained the stringency to identify those high-producing clones. So not affecting the performance of the selection process, but removing the uncertainties or the complications which MTX can sometimes bring along. With version 4, we customized the workflows. We have facilitated a more sort of adaptive workflow for the individual molecules or the individual partners which we want to work with. And we've also improved our expression vector system to include genetic elements, particularly synthetic elements, to ensure stable gene expression, as well as honing that expression within our host cell line. With the current iteration of our platform, which is version 5, we introduced a whole host of new improvements and updates to our platform, with one being that we're now able to go as fast as possible in our timeline from DNA to RCB in just 9 weeks. We included elements of automation and predictive modeling, both of which I will expand on further in the following slides. But additionally, we also increased our capabilities in genetic engineering by using our own CRISPR tool to do that. And we also developed certain workflows to support process intensification, i.e. by developing a high inoculation fed batch process, as well as also selecting clones for more of a continuous bioprocess as well. And as I mentioned with our media system, we also improved the formulation, which is in the current iteration of the SmartShow media system, where we've shown to improve the productivity as well as product quality. And all of these improvements and all of these developments are performed in our state-of-the-art facility in the south of Germany, outside a town called Ulm. And this is our center of excellence for cell line development and media development as well. So moving on to our track record. So as I mentioned, our first commercialization of our platform was around 2010, 2011. And since then, we've done over 300 different CLD projects. And we've expressed a range of different types of molecules, from your classical IgGs and monoclonal antibodies to also those more novel Fc fusions, bispecifics, as well as some of the more complex proteins which you get in the industry, such as enzymes, hormones, and cytokines as well. And what you can see is with the range of titers that we've achieved some good titers with some of those more complex molecules as well, which shows that our platform has a versatility to also express non-monoclonal antibodies and also those more difficult to express molecules. In terms of our track record with the regulators, as you can see here on the right, we've had a range of different cell lines go to different clinical phases, and particularly 7 of those cell lines have proceeded to market approval and commercialization with our different partners or customers, which shows that our platform also has regulatory compliance with the individual regulators in the different regions as well. So a little bit more about our platform and how we perform our CLD. So once we have been given the gene of interest, we'll synthesize it with a third-party partner, And once we have that DNA sequence, we would clone it into our proprietary expression vector, transfect it into our host cell line, and generate large bulk pools. Those large bulk pools are then inoculated into the nanowell, and then using the automation of the Sartorius cell selector, we're able to perform single cell cloning. And this process I'll go into a little bit more detail in the coming slides. Associated with the automation of the single cell cloning process, we've developed novel selection tools with a productivity assay, and as well to essentially screen those clones as early as possible and identify those high producers as early as possible. And then we migrate into phase 3, which is the clone expansion and pre-selection phase, where we go from 384 well up to shake flask, and in the interim also include another selection strategy where we take a subset of those clones and perform a small scale fed batch evaluation on them. And ultimately, taking all the data we've generated and evaluating the clones, we are able to narrow it down to our top 24 or 48 clones, where we then run it through an Amber 15, which is a very, very familiar process within the industry, and a more controlled cultivation methodology to really evaluate the clone performance. And through some CQA analysis, as well as growth profiles and titer profiles, we're able to take that data and narrow down, together with yourselves, the top 4 clones, which we then establish an RCB. And all of that can be performed within just 9 weeks. Once we have those top 4 clones, we'll then perform a stability study to ensure that all 4 clones are stable, and then together narrow it down to what would be your lead clone. And that lead clone would go into a 5-liter bioreactor run using our optimized upstream process design to ensure that the clone has scalability as it starts to go towards the next phase, as you go to process development and onto GMP manufacturing and scale up. And then you have your final clone delivered, essentially. So that's a snapshot of the workflow. And now I'll sort of dive into a little bit more into the digital solutions, and one of them being automation. And as I mentioned, with automation, the key added value we found with this process is that it's allowed us to accelerate our CLD process. So how have we gone about and done that? So I mentioned that with the automation, we've used the Sartorius cell selector, which is this machine here on the left. It's made up of 2 key modules. Firstly, the module on the left is a robotic arm, which uses a single-use glass capillary, which allows automated transferring of cells from plate to plate. And then on the right here, you have the microscopy unit, which takes images both in bright field and fluorescence. And the fluorescence has a key function, and I'll get to that in the following slides. But with that tool and is its own proprietary image analysis and software capabilities. And combined together is how the machine has its automation. But how is a single cell cloning actually performed? Well, that's performed using the nanowell plate, as I mentioned previously. So this 24-well plate is made, it has the 24 wells within it, and each one of these individual 24 wells has 4,000 nanowells. And those nanowells is where the transfected pools are inoculated at an optimal concentration, which then essentially sees that you'll have a distribution of cells of either no cells, 1 cell, a single cell, or multiple cells in the individual nanowells. And then through the software's capabilities of screening and scanning each of these individual nanowells and identifying those where there is a single cell, we're able to essentially perform the single cell cloning process. Now, what are the key advantages? And they're twofold with this system and this methodology. Firstly, the automation allows a higher fidelity of single cell cloning, minimizing the operator error which might come about with it. And secondly, through this methodology, what we found is an improvement in clonal outgrowth by 40 to 50%. And this can be shown by the bar chart here on the bottom when we compared it against our previous iteration using the facts. And what you can see here is in all cases, the outgrowth from the cell selector was much better. And in the case of product 3, which was a difficult to express molecule, we can see that the improvement is substantially higher than the average 40 to 50% we generally see. And why do we see this improvement in clonal outgrowth? One of the hypotheses we have is that with the nanowell system, all the cells are separated in the individual nanowells, but the media which they are submerged in is shared, which means that the cells can still perform the cell-to-cell cross-talking while still being segregated into the individual nanowells, which essentially jumpstarts them from the lag phase into exponential phase quicker. So that's one of the key advantages. The clone outgrowth essentially also accelerates the timeline towards how we can migrate from single cell cloning into clone expansion. Now, with any sort of methodology, and particularly single cell cloning, one of the key factors, and probably the most important factor in the cell line development process, is the assurance of monoclonality. And the way we assure monoclonality with this methodology is twofold. Firstly, we've developed a single cell cloning validation report, which is essentially a statistical evaluation of our single cell cloning methodology and ensure that it's in alignment with FDA recommendations. And through that, we've also calculated an average probability of monoclonality, which is 99.7%. So it's quite a robust methodology in assuring monoclonality. The second layer of assuring monoclonality is actual physical traceability of your clones. And the way we go about doing this is that when we deliver our the CLD project report, or the completion of the project and the report gathering all the data generated through the process, we will also include the physical images taken by the cell selector of the top 4 clones or for that specific project. So you'd get the image on day 0 where you see a single cell, and then on day 1 where you see 1, 2, or 4 cells that then we deem that is a cell line which has been truly derived from a single cell. And together, both of that data really shows full traceability as well as assuring monoclonality from a statistical evaluation as well. So as I mentioned, the cell selector actually also has fluorescent capabilities, and there is a key advantage which we've leveraged with that technological capability. And what we've done is we've developed a novel productivity assay, which we can perform within the first 4 days of the single cell cloning process. And the way we go about doing this is that after inoculating the cells into the nanowells and taking images on day 0 and day 1 for monoclonality, what we will then do is introduce and inoculate a protein A-coated bead and a secondary fluorescently labeled antibody. And and this is distributed across the individual nanowells. And then we incubate the plate for up to 4 days, or on to day 4, where then you have the clones secreting the antibody or molecule of interest, it being immobilized onto the bead and tagged by the fluorescently labeled antibody. And once a confluency is reached after 4 days, what we do is we take an image by bright field to measure the confluency, and then by fluorescence to measure the relative fluorescent intensity. And here you can see an example of across nine nanowells. You can see some where you see low fluorescent intensity, some with higher, and then those with none. And then this is the advantage of the software of the cell selector. It can identify and track these individual wells, and then program the robotic arm and the single-use glass capillary to transfer that selected well into your 384 well for clone expansion. And we did an internal evaluation of this assay to see if it has its advantages. And you can see this on the right here in the bar chart, that with using the fluorescent assay and identifying those wells where you see relatively high fluorescent intensity, we can essentially identify and bias our selection to identifying those high producing clones, which we might not see if we just chose random selection or general confluency as the first selection tool in the first 4 days of the single cell cloning process, and as we migrate to clone expansion. So that's another key advantage of the automation which comes with the single cell cloning methodology. So how has that overall accelerated our timelines? Well, with our previous iteration where we used the FACS, we would have our vector, we would transfect it into our host cell line, and we'd establish mini pools and evaluate those mini pools in a fed batch evaluation, and essentially choose a subset of those mini pools to then go into single cell cloning to ensure better recovery of those of the individual clones once we go through the FACS system. And ultimately that would take at our fastest iteration then 14 weeks to achieve your top 4 clones and establish the RCB. Whereas currently with the implementation of automation and the cell selector, we're able to again transfect, generate pools, but rather than generate mini pools, generate bulk pools and goes directly into single cell cloning, where then due to the improved clone outgrowth, as well as interim selection strategies and a more robust clone selection strategy using the small scale fed batch, using the predictive model, which I'll touch on next, we're able to ensure that we get those high producing clones and do it faster in just 9 weeks. And this is a saving of approximately 5 weeks, which then, can influence the overall CMC process even further. So that's about how we've been able to accelerate the process using automation and using the strengths of the cell selector. The next element, of which I wanted to talk about, is the predictive model using machine learning and how it's helped us to de-risk our CLD program. And the way we've gone about doing that is, firstly, the goal of the model and the predictive model using machine learning was to essentially use the data from the cell selector to predict clone performance in the AMBER 15 run, which is the final evaluation you would do to narrow down your top 4 clones. And the key challenge we wanted to address in this model is that we wanted the model to be able to predict those high producing clones when it's product agnostic. And what I mean by that is that the different projects would express a different molecule, right, depending on the client or our partner, which molecule they'd like to express. But we wanted the model to work irrelevant of whichever molecule was being expressed, and still be able to differentiate between high producers and low producers. And so in a summary, how did we go about building this model? And this can be summarized on the slide here at the bottom, where we started with data collection from the cell selector, and then we went through a pre-processing phase where we took data from 5 different products, the raw data, we normalized it to allow it to be more directly comparable and make a more representative data set. And then we implemented a multi-model approach using PCA, using artificial neural network classifiers and random forest, where the latter two are machine learning algorithms. And together, this multi-model was merged into a single output, giving you a ranking. And I will now kind of sort of break down each one of these individual steps a little bit more and sort of delve into a little bit more detail of how this digital solution has supported us in de-risking the program. So firstly, the data. So as I mentioned, the cell selector had its strengths in sort of accelerating the process, but actually it also had a strength in capturing a lot more data from it. And the key factor of how it does this is that it's imaging software and it's image analysis software. So as I mentioned, the cell selector takes images of the nanowells, and as you can see here, between a high producer and a low producer, it has a single cell. You can see the fluorescent intensity or the area of fluorescent intensity. It can identify the confluency and the outgrowth, but it converts these images and the data from these images into a whole host of different parameters and numerical parameters for the cell or fluorescence, which includes shape factor, diameter of the cell, sphericity, etc. And also in terms of fluorescence, your number of found particles, your fluorescent, the total area of detection, intensity, etc. So there's a whole host of over 80 different parameters which the cell selector can capture in a numerical value. So that's a lot more data which we can generate from a single cell cloning process than just clonal outgrowth and fluorescence. So this data is what we can then use to plug into the model. But one of the factors we needed to keep in mind in this process is that, firstly, that the process essentially uses all this data, but there is also variation between project to project. So one of the things that the advantage of the software is that it has project to project variation is minimized because of its auto-focusing capabilities and the software itself. And then combined with our pre-processing and normalization of the data, we're able to essentially merge it together and make it a more representative data set. But when going through this process, one of the key factors we found is that there was actually a key challenge we needed to address. And that is, between the cell selector and the AMBER 15, the cultivation conditions are very different. So for instance, in the cell selector, you can see the medium is different in the form of a cloning medium, whereas in the AMBER 15, we use a production medium. In the terms of the cell status, in the cell selector, they're separated single cells and they're still going through recovery, whereas in the AMBER 15, it's a fully recovered cell or a cell line. In terms of cloning condition, oh, well, not cloning conditions, sorry. In terms of cultivation conditions, it's uncontrolled, whereas in the Amber 15, it's a more controlled condition. And then in terms of cultivation, there's no feeding and the focus is on outgrowth, whereas in the Amber 15, there's a regular feed and there's a focus on production. So the two models are very different. The cultivation models are very different. So when it came to predicting from the cell selected data what would be clone performance and trying to predict absolute titers, and factoring in also the biological complexities of different products and also individual clones having different methodologies, it becomes quite a challenging was coming quite challenging and quite complicated, and the predictive strength was not very strong. So what was the solution to that? So what we identified was that, and this can be shown here on the graph on the right, where you can see the predictive line, where you'd have absolute prediction or 100% accuracy on your prediction. You can see that in the with high producers or higher titer clones, they do deviate away, and lower producers, you do see some deviations or further away from the predictive accuracy. So, but what we could do instead is, what if we set a threshold? So we set a threshold and identify in a binary way what would be classified as a high producer and a low producer. So in this case, you can see in the turquoise boxes where if you set a threshold of approximately 2.5 grams per liter, you can identify those high producers as above 2.5 grams per liter, and those which lower than 2.5 grams per liter as a low producer. And suddenly then through the classifiers, you're starting to capture more accurate data or more indicative performance of those which are high producers and lower producers, and then the predictive strength starts to improve, and you're able to factor in the complexities, but then still be able to have an early indication of a high producer and a low producer. And the reason we chose to do this rather than try and take data from a later stage in the in the clone expansion process is that we wanted to de-risk the program as early as possible. And this is one way of doing it, is to get that really as early as possible. So within the first 4 days, an indication of what would be a high producer and a low producer, and then be able to really bias that selection moving forward. So after addressing that challenge, what we went about doing is having this normalized data. We've addressed the challenge and gone with a more classifier approach of identifying between a high producer and a low producer. We then developed the multi-model approach. And what that entailed is taking a PCA classification model and then combining it with a artificial neural network classifier and a random forest classifier, both of which are machine learning algorithms, and be able to essentially plug the data into each of these individual models and use essentially a multivariant data analysis tool, in this case, SIMCA, which is Sartorius' proprietary solution for that, be able to then essentially predict where a high producer from these three models, and then be able to integrate that into the software and have a one button click clone ranking. And the way the ranking would work is essentially, if all three models identified a clone as a high producer, it would be the highest, it would be at the top, and then at the bottom of your rank would be where all three models predicted the clone would be a low producer, and that would be ranked at the bottom. And then you get the gradient of all the different models as it ranks all your clones individually. And essentially what we've been able to do is merge that into it and be able to generate a model. Now, a key aspect to the model is, how do you keep improving the model? So every 4 to 6 months, with the new data we generate, we will then plug that back into the model to strengthen the prediction of every project which has been done. Additionally, what we've done through developing this model and screening all those 80 different parameters, we've been able to essentially narrow it down to what are the key cell parameters that essentially allow you to determine high producers and differentiate them from those low producers. So that's how we've done the model. But how does it work? So we did an internal evaluation, as you always have to do when you're developing something like this, and try and tested it on 2 different products. And what you can see here on the data on the right here is that with product 1, we saw on average, we saw more than 40% increase in average titer of the top 4 clones, and that was statistically significant. And this can be highlighted by the clones, the top 4 clones highlighted in pink. And how we compared that is using the productivity assay with the cell selector, so really measuring clonal outgrowth and then the fluorescent intensity, and combining it then doing that with the combination of the model, where you're capturing a whole range of other parameters which the cell selector can factor in, and using the predictive model and the machine learning algorithms to give you that indication. And similarly with product 2, what we saw is that more than a 60% increase in average titer of the top 4 clones. And there, essentially, you can see here that a couple of clones you may have not even identified if it wasn't for the advantage of the model. And that was using the same sort of comparable between just clonal outgrowth and fluorescent intensity in the productivity only assay, and then using the model on top of that as well. Now, having a model which gives you good indication is one thing, but having a model which is robust is also another very important part, because one of the key factors of being able to de-risk your program is making sure that then with the robustness, you're not relying on a numbers game of probability to identify the high producing clone, but rather more of a more mathematical and methodological approach, and a reliable and robust approach to achieve that. So how we went about assessing the robustness of our model, and particularly the robustness of our multi-model, where we have 3 models in one, is by essentially comparing between a low confidence and a high confidence. And what I mean by low confidence and high confidence is that where high confidence was where the 3 models which I defined all indicated, or 2 out of 3 of the models indicated, that this clone would be a high producer, and compared it against where only one of the models identified the clone would be a high producer, and differentiating whether if all three or 2 out of 3 indicated that it was a high producer, whether that would be more effective. And what we can see here on the right is that we saw more than 50% increase in the average titer of the top 4 clones using the high confidence multi-model approach compared to the low confidence. So 2 out of 3 or 3 out of 3 of the models predicted it was a high producer, had a more effective predictive power and a stronger predictive power than those which were using the low confidence. So ultimately, what that means is that using this multi-model approach, being able to capture more data from the cell selector and using that to strengthen our selection strategy, we're able to de-risk your program to identify those high producers as early as possible, that then when we go through the clone expansion process to narrow it down to your top 24 and eventually to your top 4, we already have good indication from the beginning of how that performance will be. So in conclusion, what does our platform, what digital solutions have done, and the 2 key digital solutions have helped in improving our platform is through automation, we've been able to sort of improve the single cell cloning process, capture more phenotypic data on the clones, and then combining it with the predictive model using machine learning, we've been able to facilitate an early prediction of those high producer clones. And combining these together, we've been able to essentially accelerate the CLD process by automation, so going from DNA to RCB in 9 weeks, and also at the same time de-risk your CLD program by having a high fidelity single cell cloning methodology, improving the clonal outgrowth, and then ultimately using the model to get early indication or early identification of those high producers to ensure that you get the optimal clone at the end of the CLD process. And the advantage of this and our platform, as I mentioned before, the versatility, is that this model applies to mAbs and mAb-like molecules such as bispecifics, multispecifics, Fc fusions, and also biosimilars. And with that, thank you very much for your attention. Happy to answer any questions you might have.
All right. So thank you, Dr. Yash, for the very insightful presentation. I hope the audience have learned a lot from the valuable sharing session on how they can digitize their cell line development process using Sartorius platforms. So once again, attendees, if you do have any questions regarding the presentation, please submit your questions by using the Q&A function at the bottom of your screen. And now we'll proceed to the live Q&A session. So along with Dr. Yash, we are glad to have Mr. J. Kumar Barrett, Manager of Product Excellence, and Ms. Judy Huang, Manager of Sales Development Specialist, also from Sartorius, with us to answer your questions. Here's a bit about J. Kumar and Judy. Jaykumar Barrett is an accomplished upstream process scientist with over 17 years of experience in the biopharmaceutical industry. He specializes in the development and optimization of biotherapeutic molecules with a strong focus on biosimilars. Jayakumar's expertise includes process development, optimization, scale-up, and technology transfer, making him a key contributor to advancing bioprocessing solutions. Meanwhile, Judy Huang is a biopharmaceutical professional with nearly a decade of experience in the industry. She is currently the manager of sales development specialist at Sartorius. Her technical expertise centers on mammalian cell cultures, medium formulation design, and bioreactor operation, supporting her commitment to delivering innovative solutions in biomanufacturing. So now let's look at the questions we have. We have a few questions already coming in. So the first question, I'll read it out loud to Dr. Yash. There's two parts to this question. So the question reads, what is the selection marker of Sartorius cell line? And what is the strategy to perform stress induced during single cell cloning?
Sure, that's a good question. So our selection system is actually DHFR. So we use excuse me. So we use DHFR as a selection methodology. It's quite, as I mentioned, it's quite a popular or quite a well-known selection system within the industry for generating cell lines. And essentially, it's a metabolic selection system, which is fundamental to the survival of the cell. And therefore, the host cell line is deficient of this DHFR gene. And by recombinantly expressing it in parallel to your gene of interest, we're able to essentially identify those cells which have the integrated genes of interest. So say your heavy and light chain for an antibody, as an example, which is concurrently expressing this recombinant version of the DHFR gene, which allows us to select those cells. And in terms of stress induced and how do we select, or how do we essentially get the stringency of selection? Well, as I mentioned, we removed MTX from the process, which was the initial way to identify those high producers from a CHO cell line. Instead, we engineered our vector to have a way to differentiate a very low recombinant expression of that selection gene, and therefore only cells which have been selected that have a high enough threshold and concurrently then being able to also express your heavy and light chain or your molecule at the highest level would then be able to be selected. So the way we've adjusted the stringency of selection has come by removing MTX, but being able to maintain it using engineering of our vector system specifically. And so that's how we essentially select our cells.
All right. Thank you, Dr. Yash. And moving on to the next question, once again by an anonymous attendee. Can Sartorius offer gene editing service? How long will it take to get a RCB?
Yeah, so we do offer gene editing services. I kind of touched on it at the very early introduction phase, but essentially we have developed our own sort of CRISPR tool. And as a proof of concept, the first way we've done that is we've developed a FUT8 knockout. So a very common process within the industry. So FUT8, a knockout essentially is the knockout of a FUT8 gene, which essentially allows your cell line to express a molecule without fucose. So it's afucosylated. And the way we've gone about doing that is using our gene editing tool, and we actually knock it out in parallel to expressing the molecule or integrating the gene of the molecule of interest, say an antibody in this case. And so we do that in parallel, and all it does is adds an additional week to the timeline. But where we're doing the single cell cloning and the clone expansion, we also do the selection of clones by two criteria. It's expressing your molecule of interest, but it is also demonstrating the knockout of the gene of interest by twofold, both by flow cytometry as well as sequencing.
All right. Thank you, Dr. Yash. Moving on to the next question. It's from Angelo Brani. It reads, do you have a CLG process for more complex selection like biosimilars, not only looking for productivity, but also for glycans, charge, variant stability, and other necessary quality attributes?
So that's a good question. Yeah, fully appreciate with biosimilars, it isn't just down to growth and titer, but actually some of the additional sort of CQAs which you necessarily need to look at for biosimilarity. In that case, what we go about doing is that the digital solution and the automation, the initial selection of clones starts with starts with just growth and titer. As I demonstrated with the cell selector, we can express we can get a relative indication of high producers by fluorescent intensity. But once we get into the clone expansion process, we start to then use a small scale fed batch, again, still to evaluate titer and growth. But where we then want to identify those clones, particularly for CQAs, is at the Amber 15 level. So what we do there, particularly for biosimilar, we do increase the sample size of clones which we screen. So there, where I mentioned we do 24 or 48, we would use our larger approach of taking 48 clones, culturing them in the Amber 15 to generate enough material to then do more sort of extensive analysis in terms of CQA. So looking at N-glycan profile, looking at charge variant stability, and other quality attributes, particularly also sometimes binding to your target antigen, and also sometimes potency assays where necessary to determine biosimilarity as early as possible. So that's how we go about doing when it comes to developing biosimilar molecules.
All right. Thank you, Dr. Yash. And moving on, the next question is by Shreyas. It reads, do you have any commercially available prediction models for someone who has a cell selector?
Very good question. At the moment, the model was our innovation. We've developed it in-house. So it is specifically for our cell line, a CHO DG44 cell line. In terms of commercially available, that is something which we're investigating in the longer term future, particularly maybe more as a collaboration rather than a commercially available tool. Maybe Judy or Jay, if you have any other additional thoughts on that.
Yeah, so as Yash rightly said that currently this model is for our own internal cell line only, but definitely please reach out to your BioA representative or colleagues from Sartorius, and definitely we can consider a particular request on case-by-case basis, and we can collaborate, so we can work on more of a collaborative model than a commercial model.
Okay. Yeah. Thank you, Dr. Yash and Jay. So moving on, we have the next question, which reads, what advantage does cell selector offer over conventional single cell cloning methods?
Good question. So the cell selector, I kind of touched on it a little bit. So there are a range of different conventional single cell cloning methodologies. So what I will do is I'll compare it to our experiences specifically and to the FACS, which was our previous iteration, which was also a very common technique used in the past for single cell cloning. The advantage is, firstly, automation. It allows us to get higher fidelity when it comes to the single cell cloning process. Also, based on using the nanowell plate, one of the other ways we've been able to sort of see the advantages of the cell selector is that the ratio of the imaging area. So your nanowell is a much more smaller area to scan rather than a 384 well plate diameter. And so relatively your size of your cell to the area you're scanning by the microscopy unit is much easier to identify those cells and not eliminate them to maybe debris or any other sort of other elemental particles which might distract it away from it being a cell or make it a questionable well. So that's another aspect. But I think one of the strongest advantages is the recovery, which I mentioned, where on average we see 40 to 50% improvement in clonal outgrowth. And as I mentioned, it's a range of reasons for it. One of them being that the cells are allowed to crosstalk because they're still submerged in the same sort of media system while being separated by the nanowells. And so that allows the cells to jump into exponential phase quicker. Another element is using the single-use glass capillary. We found that has it's not as stressful on the cells when it's going from the transferring process, so the cell is not disrupted as it goes into the 384 well plate as well. So that's another advantage, is that there's less shear forces when you're transferring the cells when they're at a very temperamental stage. So that's another advantage.
All right. Thank you, Dr. Yash, once again. So moving on, we have another question from Shreyas. It reads, what is the regulatory acceptance of using fluorescence beads antibodies to check productivity?
That's a good question, and one we've had a few come up a few times. So, yeah, we fully appreciate that. You are using a labeling antibody and a bead into the single cell cloning process. And what we did is we had to do a risk analysis for this and align it with what is required by regulatory agencies. And what we found is twofold. Firstly, both the bead and the secondary fluorescently labeled antibody adhere to the TSE/BSE requirements when it comes to your RCB, or essentially when you're going through the single cell cloning process. And so therefore, based on the TSE/BSE evaluation and also the certification, the risk of animal components or any sort of undesired components in your cell line is already being factored in. And that is in alignment with regulatory requirements. So we're not breaching that or adding undue risk when it comes to filing. Secondly, with the bead and the fluorescently labeled antibody, as we go in through the clone expansion process, it is constantly being diluted. So by the time you establish your RCB on the top 4 clones, the dilution factor is so large on the number of culturing and subculturing you do through the process that actually we found, based on our risk evaluation, the traces of it is negligible. So it's twofold. Firstly, how the materials are sourced, they are in alignment with the regulatory requirements in terms of component composition of it. And secondly, it is also negligible because it is also diluted out over the expansion process.
Okay, thank you, Dr. Yash. Anything to add by Jay or Judy? Okay, I think it's all clear. Moving on, we have a few more questions coming in. So the next question reads, it's by Arindam. The next question reads, in your bead-based secretion assay, beads are visible along with cells in the microscopic image, making it difficult to distinguish cells from beads, eventually affecting the monoclonality report. Dr. Yash, any comments on this?
So, yeah, it's a good question. And what I would say there is actually that's the strength of the software. So you're right, you would see the beads at least in the first 4 days. But what I would say is that the monoclonality assurance report is already taken before the beads are added. So what I mean by that is that we take the images for traceability on the day 0, so the day of inoculation, and then day 1, where we have either 1, 2, or 4 cells. So that is the images which will be provided, and that is the images necessary for assuring monoclonality when it comes to filing. So you're doing your IND filings and assuring your cell line is actually derived from a single cell. So yes, once the bead is added, you can visibly see it in the microscopy, but those images are not necessarily needed because to assure monoclonality, it's day 0 and day 1 after inoculation. I mean, Jay, Judy, anything to add to that one?
Yeah, and the other fact is that the size of the beads. So the size of the beads is only 4 to 4.9 micron. So it is definitely very smaller than the cells. And our software is trained to recognize, because it's a fluorescent based, so beads will be a fluorescent beads. So that's where the recognition will happen. So the software is very well trained, and as Yash already mentioned, that the monoclonality report images are actually well taken before the beads are added.
All right. Thank you, Dr. Yash and Jay. So moving on, we have the next question. The question reads, if I send data from cell selector to Sartorius, can I request the use of SIMCA machine learning program, or is it possible to use the SIMCA program online?
Also a good question. Clearly some interest in our model and how we've set it up. What I would say is, as I kind of touched on a moment ago earlier, and I think maybe I'll also hand it over to Jay and Judy about this one from a commercial perspective, but from a practical perspective, I think it would be more of a collaborating approach. So it would be one where maybe we could take that data and work with our data scientists in our team who have developed the model, and maybe also discuss a little bit more about what cell line, what it was used, so that we can see how applicable the model would be compared to how we've established it with our cell line. But from a commercial perspective, Jay, Judy?
Your turn, Judy, maybe.
Can you hear me?
Yeah.
Yes, loud
Okay,
and clear.
yeah. So, of course, we can offer the digital solution for every customer. If you are interested in the implemented into the cell line development, we are happy to support. And if there's any consultant request, as you see that we have Dr. Yash and also the full scientist team to make it happen, and you will get the digital solution implemented into your platform for sure. Yeah.
Okay, great. Thank you, Judy and Dr. Yash. So moving on, we have another question by an anonymous attendee. It reads, does the model and selection process also predict clone stability? Or is the technique to remove MTX already leads to increased stability in the cell line?
Good question. So the model doesn't take into consideration stability of the clone, and part of the reason is that the model was already complex enough when trying to predict sort of expression and it being molecule or your protein which you're expressing being independent of that or agnostic. So the stability we sort of have already developed over the iterations from version 1 to 5 now, and through the MTX approach. So as the attendee kind of mentioned, it is we're more confident in our stability because of our past history and how that cell line is developed. We've actually done it more, the stability is more done on a genetic level in the sense of including necessary elements in the vector system, which allows us to essentially maintain a stable transcriptional activity as well as ultimately protein expression from it. And also the addressing of the MTX and removing that has given us more confidence in our stability. So yeah, to answer your question directly, the model does not address stability, but rather we've used MTX and other genetic elements within our vector to ensure that. And what we've generally seen in our stability is that on average, we see around 81% stability out of the 4 clones we evaluate. So really, majority of the time, we see all 4 of them are there, but on average, over a subset of projects, we saw on average 81% stability. So quite robust.
All right, thank you, Dr. Yash, once again. And moving on, we have another question. It's by Praveen. It reads, by clone selector, based on ranking it, will select and what parameters it will be selected, high secretory clone?
It's a good question. So as I mentioned, when the cell selector takes the image, it converts them into a range of different numerical values. And those are the 80 different parameters, both on the cell and on the fluorescent side. What we did through the model is evaluate all of those different parameters, and we found what is the subset of actually indicative parameters to identify high producers over low producers. Unfortunately, I can't divulge for proprietary reasons what those parameters are in this case, as it's kind of part of our model and the sort of the IP around it. But rest assured that it is some of the key parameters you would generally indicate for high secretory clones.
All right, thank you, Dr. Yash. And due to time constraints, we'll go for just one final question. So there's a question from an anonymous attendee, and it reads, is there any IP concern while applying CRISPR tool?
Also a good question. As we know that landscape is a bit complex at the moment, particularly with how IP and patenting is linked with CRISPR tools. So what we've done is that we have developed or improved on a platform which we've licensed in, but in terms of IP, we have the freedom to operate through our platform. So through co-developing with a partner, we have been able to essentially have the freedom to operate on our CRISPR tool with our cell lines, while and still be able to and not have any sort of complex IP or royalties or licensing fees associated when generating a cell line with that approach.
All right. Thank you, Dr. Yash. And I think that wraps up our Q&A session for this webinar. So thank you, Dr. Yash, Judy, and Jay Kumar for clarifying the doubts of our attendees for the day. As much as we would like to address all questions from the attendees, in the interest of time, we will have to wrap up today's session. Not to worry, any unanswered questions will be submitted to our speakers, which will be answered offline and shared with all attendees via email. I hope you all enjoyed being a part of this webinar and found the presentation and discussions very insightful. IMAPAC would like to extend our heartfelt gratitude to Sartorius for making this webinar possible, and a big thanks to the audience for being a part of and making this webinar a success. Again, on behalf of Sartorius, thank you, and looking forward to welcoming you again at the next webinar.